Assalamualaikum and hi. Today we are going to present about our cell mini project, which consists of two experiments, which is experiment 1 redox reaction and catalyst activity, and experiment 2 is cellular respiration.
Abstract and introduction for experiment 1. Redox reaction and catalyst activity play pivotal roles in driving efficient and environmentally friendly chemical transformation. This experiment examines the redox reactions and catalyst activity on living tissue, different types of tissue with hydrogen peroxide to observe catalyst activity, and different types of tissue with air, lime juice, and tap water to observe redox reaction. Redox reaction short of reduction oxidation reaction, which involve electron transfer and play a crucial role in diverse fields. In biology, catalyst, a vital enzyme, exemplifies by this facilitating the breakdown of hydrogen peroxide. This process showcases the interplay of redox reaction in cellular homeostasis, emphasizing the importance of understanding this dynamic relationship for insight into both biological system and broader application.
This experiment found observing redox reaction. The apple was cut into three small pieces and then put in a different wash place, which was exposed to air, water, and lime juice. And then the color changes was observed and recorded. Last thing, which is the catalyst activity, we need to use hydrogen peroxide that was put into 2 ml into the different test tubes that was labeled into A into B and was cut and weighed the 2 grams of apple. And then we need to observe and record the rate of the catalyst activity after 10 minutes. Then class 3, which is the effect of the temperature, we need to use 3 different temperatures into the 3 test tubes, and then we put the chicken liver that was cut and weighed, and then we need to observe and record the observation.
On to the result of experiment 1 by A, which is observation of redox reaction. We were able to observe that slices of potato and apple that were exposed to the air were the most visible color change, where it became darker yellow in color. Meanwhile, slices exposed to water have only slight change in color. For slices exposed to the lime water, it have the lightest change in color, which it appears to be same as initial. The color changes happen due to the enzymatic browning, when enzyme polyphenol oxidase produce all quinones that reacts with amino acid to produce brown melanin. Meanwhile, slices were coated with lemon juice acid with high pH and antioxidant that inhibit the oxidation reaction.
So as you can see here, the result for part B, which is catalyst activity experiment, basically for the initial height, it all started with 0 centimeter. For the final, the highest bubble that produced by cell is chicken liver, which is 4 centimeter, whereas the least produced bubble is soap piece, which is 0.6 centimeter.
This is the result of part C, which is the effect of temperature on catalyst activity of liver at 0 Celsius. Bubble form is 0.3 cm. Meanwhile, at 24 Celsius, the height of bubble form is 1 cm. Lastly, at 95 Celsius, the whole bubble will form. This shows that the optimal enzyme to operate is around 25 Celsius. Meanwhile, the enzyme are denatured. Thus, were not able to undergo cellular respiration due to high temperature at 95 Celsius.
So for the conclusion, the redox reaction experiment was successfully conducted because we could observe the color changes before and after of the cell. Whereas for catalyst activity, we managed to conduct the experiment successfully because we were able to collect the height of bubble produced. The hydrogen peroxide is broken down into water and oxygen gas when all the living tissues reacted with the catalyst enzyme in the damaged cell.
Abstract and introduction for experiment 2. This experiment used methylene blue to observe cellular respiration in yeast of different sugar. Head of bubbles of yeast suspension were recorded to observe the absorption of cellular respiration that occurs in yeast. Cellular respiration is the process by which cells extract energy from nutrients like glucose to ATP. The energy currency for cellular functions is essential pathway occur with or without oxygen, playing a central role in sustaining cellular function.
Experimental cellular respiration, we use 20% of glucose, lactose, sucrose, maltose in the different test tube that has some distribution of the yeast. And after 15 minutes, we use methylene blue as an indicator to record and observe the changes further.
For experiment 2, which is cellular respiration, the results concluded that the glucose have the higher rate of bubble formation, which is 2.6 cm, than maltose, sucrose, and lactose. Then the formation of bubble in sucrose is 1.4 cm, while maltose and lactose has the lowest rate of bubble formation, and distilled water acts as a control group. Glucose is a monosaccharide, which is a simple sugar that has simple structure and less bonds to break. That is why glucose has the higher rate of bubble formation. In conclusion, the cellular respiration in yeast were able to be observed by the bubble formation on top of the yeast suspension in glucose, sucrose, and maltose sugars, which indicates that carbon dioxide CO2 were produced by the process of enzyme breaking down the sugars. That's all from us. Thank you.